Mitosis. Mitosis refers to nuclear division in body cells. You remember that every cell goes through a cell cycle. In this cycle, much of the time is spent at interphase, which is the non-dividing stage. At late interphase, two things happen to prepare the cell for nuclear division. Do you remember what these two things are? Well, they are the replication of DNA and the division of centrioles. So after the DNA has replicated and the centrioles have divided, the cell is now ready for nuclear division. In body cells, nuclear division occurs through a process called mitosis. Mitosis can be divided into 4 stages: prophase, metaphase, anaphase, and telophase. It is important to note that the whole process of mitosis is continuous, and the cell does not stop at any one of these stages. Let's look at the cell cycle again. Notice that the nuclear division portion is divided into 4 sectors. Now we know that each of these represent one of the stages of mitosis. This is prophase, metaphase, anaphase, and telophase. Now we shall look at each of these stages in greater detail. Firstly, prophase. During prophase, condensation of chromatin threads to form chromosomes occur. What does it mean by condensation of chromatin threads? Now, this is how the chromatin threads look like in the cell at interphase. They are very, very long and thin, barely visible. This diagram shows the chromatin thread which has replicated, so you see 2 sister chromatids joined at the centromere. Now, during prophase, these sister chromatids will start to condense. That means they will start to wind upon themselves, like this, wind upon themselves and become shorter and thicker to form a chromosome. So a chromosome is actually thicker and shorter version of the sister chromatids of the chromatin thread. So these are the 2 sister chromatids joined at the centromere. These 2 sister chromatids, if you remember, they are actually identical because one was copied from the other. So this is one chromosome. So when condensation of chromatin threads occur, they produce the chromosomes. So during prophase, the chromosomes start to appear inside the nucleus. Each chromosome will be seen as a pair of sister chromatids joined at the centromere. So this diagram shows the nucleus having 1, 2, 3, 4 chromosomes. Besides the condensation of chromatin threads, the centrioles that have divided during interphase also start to produce microtubules that radiate out from them, forming esters. Now, the 2 pairs of centrioles then start to move towards opposite ends of the cell. This pair of centrioles will move to this end, and this pair of centrioles will move to the other end, like this. Right? So what happens is when they move to the opposite ends of the cell, they have spindle fibers that stretch from the centrioles at one end to the centrioles at the other end. These spindle fibers form what is called a spindle. By now, the nuclear membrane has disintegrated and the chromosomes are freely floating in the cytoplasm. This picture shows how the cell look like at prophase. Notice that there is no nuclear membrane and you see a lot of chromosomes inside. After prophase is the next stage called metaphase. At metaphase, those chromosomes that were freely floating around in the cytoplasm start to align themselves at the middle part of the spindle. Now, this middle part of the spindle is called the equator. So each chromosome will be attached to a spindle fiber by the centromere, and they're all arranged at the equator. It is easy to remember M, metaphase. At this stage, where are the chromosomes? They are at the middle, M, middle of the cell called the equator. So this picture shows how a cell looks like in metaphase. Notice that the chromosomes are all at the middle of the cell. After metaphase comes the next stage called anaphase. During anaphase, these spindle fibers start to contract. So when they contract, they will pull. You look at this, if these spindle fibers contract, this one contract, it will pull at the centromere. This part also contract, it will pull at the centromere. So during anaphase, contraction of the spindle fibers actually cause the centromere to split, and the two sister chromatids are then pulled towards opposite poles. So anaphase is marked by the pulling apart of sister chromatids. So again, it's easy to remember anaphase. Anaphase A, what happens to the chromosomes? Well, the sister chromatids are pulled apart. A, pulling apart of the sister chromatids. Now, the moment these sister chromatids are pulled apart, they now are called daughter chromosomes. So each of these is now a chromosome, meaning there are actually now 8 chromosomes inside the cell. This is how the cell looks like at anaphase. Notice 2 groups of chromosomes moving towards opposite pole. Yeah. So when the daughter chromosomes are pulled towards opposite pole, at the end of anaphase, we will see one group of chromosomes here and one group of chromosomes at the other side. Now notice that these 2 are identical. Similarly, these 2 daughter chromosomes are identical. These 2 are identical. And these 2 are identical. So what do we have? We actually have 2 groups of chromosomes which are genetically identical at both ends. Now, towards the end of anaphase is telophase, right? At telophase, what happens is the nuclear membrane will start to reappear surrounding the group of chromosomes at the 2 poles, and the spindle fibers disappear. So this is what telophase look like. You know, the spindle fibers disappear, nuclear membrane begin to surround the 2 groups of chromosomes at the 2 poles. Once the nuclear membrane reappear, these chromosomes will start to uncoil and become invisible again. They become chromatin threads again. The spindle fiber breaks down. So this is how the cell looks like at telophase. You see 2 nuclei inside one cell, right? So if you see 2 nuclei inside one cell, that means nuclear division is over. So telophase is the last stage of mitosis. See, telophase. The nucleus has divided into 2. So the next thing that follows will be cytoplasm division or cytokinesis. So cytokinesis refers to the splitting of the cytoplasm. So in animal cells, it occurs through a constriction of the cytoplasm. The nuclear membrane starts to constrict, and eventually 2 daughter cells will be produced. These two daughter cells are genetically identical. Cytokinesis in plant cells is slightly different. In plant cells, a cell plate is formed between the two nuclei. And then cell wall will be deposited on either side of the cell plate to divide the cytoplasm into two. You can see that this is a plant cell because it has a cellulose cell wall. So in plant cell, a cell plate is formed, followed by cell wall being produced on either side of the cell plate to divide the cytoplasm, and you get two cells. So this is a summary of all the 4 stages of mitosis. This is prophase. What happens at prophase? Let's recap. We have shortening of chromatin threads to form chromosomes. So chromosomes appear. We have centrioles moving towards opposite pole, with esters being formed and spindle fibers stretching from centrioles at one end to centrioles at the other end. And then at late prophase, cell membrane breaks down, the spindle is formed, and the chromosomes are freely floating in the cytoplasm. After prophase is metaphase, M, where the chromosomes are arranged in the middle at the equator. Each chromosome has its centromere attached to a spindle fiber. At anaphase, the spindle fibers shorten and pull at the centromere. The centromere split, the 2 sister chromatids are pulled apart and they become daughter chromosomes. The daughter chromosomes are pulled towards opposite poles by the shortening spindle fibers. So when the daughter chromosomes reach the poles, a nuclear membrane will reappear around each group of chromosomes. And then the chromosomes uncoil to become chromatin threads again. By now, the spindle fiber has disappeared. Mitosis is over because 2 nuclei are formed. So what follows next is cytokinesis, whereby the cytoplasm split to divide the cell into 2. So from one cell, you end up with 2 daughter cells. These 2 daughter cells are genetically identical. Mitosis in plants occur usually at the root tip and the shoot tip, where the cells are actively dividing. This diagram shows the root tip. Notice all these cells are darkly stained because the nucleus has so much DNA material after the replication of DNA. So if you zoom in on the cells, you can see cells at different stages of cell division. You see this cell has chromosomes that are visible. Some of these cells have chromosomes that are visible. So root tip, shoot tip, this is where the growth occurs and the cells are actively dividing. Okay, mitosis in plant cells. Now this is a cell at prophase, at interphase. At interphase, you cannot see any chromosomes because the chromatin threads are very, very fine and they are not condensed and the nuclear membrane is intact, right? Okay, so this cell is at interphase, not dividing yet. Whereas this cell is at prophase. Prophase, we see that the nuclear membrane has disappeared. You don't have a nuclear membrane like this one, you see. And then the chromosomes are freely floating in the cytoplasm. At metaphase, M, the chromosomes are all at the middle, at the equator of the cell, like that, okay? And then this cell is at anaphase, with the daughter chromosomes moving apart to opposite poles. And this cell is at telophase. How do I know it's telophase? I see 2 nuclei inside the cell. This is the last stage of cell division. I see actually a cell plate forming here. That means cytokinesis is taking place already. right? So sometimes in diagrams, we need to be able to identify the cell at different stages of mitosis. Look at this. Identify the stages of cell cycle. Prophase. Prophase, probably this one. You see a nucleus, right? It's non-dividing, this prophase, prophase. No, sorry, I mean these are interphase, interphase, interphase. You don't see chromosomes. Then at prophase, the chromosomes appear. This cell is a prophase. You see chromosomes. You don't see nuclear membrane. Prophase, metaphase at the middle. This is metaphase, right? The chromosomes are at the middle of the cell. Anaphase, moving apart, anaphase. Now this cell is an anaphase. The sister chromatids are pulled apart. Telophase, do we see a cell at telophase? Telophase, you should see two nuclei inside one cell. I don't think I could see that here. Yeah. Okay, so be able to identify the cells at different stages of cell division. So what is the importance of mitosis? Firstly, mitosis allows sexually reproducing organisms to develop from a single cell into an adult multicellular organism. So mitosis produces new cells, right? These new cells can be used for repair and replacement of worn-out cells, or it can be used for the growth of the organism. In organisms that reproduce asexually, mitosis produces new cells for the formation of offspring.